In the previous lectures, we've talked about some aspects of the general sensory system, touch and pain. In this next talk, I want to focus on one of the special senses of which we are very aware and it forms a great part of our lives for most of us, which is the sense of vision. I want to begin by reminding you that a key aspect is that, as with the other sensory systems, the eye and the visual system are not providing us with an absolute measure. The eye is not a camera. The visual image that is received by the eye is detected and broken down into constituent elements and then reconstructed and interpreted through different stages of sensory processing and in the context of our own conscious awareness and our memory and so on. This is an amazingly complex set of processes, and we're only going to have time to consider some high-level principles. As part of that, this next slide, just to remind you about the eye not being a camera, gives us insights into the sensory processing. There are two images here that I introduced in the first lecture, the kinesia triangle and the Necker cube, both of which are classic visual illusions. In the final image, what you can see is a grey square, and that grey square is then inserted into a much darker black square or a slightly lighter grey square. The small grey square appears to be different shades depending on its surroundings. But it's the same grey, irrespective. And that illustrates the point that the visual system is operating on a contrast basis. And we're looking at differences between different areas of illumination rather than absolute levels. So, how does this work? How do we process that information? The third slide shows us a classic illustration of the eye. Light enters the eye via the cornea, which is the first stage of, if you like, processing of the image, because the cornea refracts or forms the first focusing of the image as it enters the eye. Beyond the cornea is the lens, which is a flexible structure, and that allows us to adapt our focusing to bring the image to a sharp focus on the back of the eye where we have the sensory receptors. So depending on the distance the object is from the eye, the lens will adopt a different shape. Images that are distant towards infinity, the lens will be relatively flat and have low refractive power. Images that are brought close to the eye, the lens becomes fatter and increases its focusing power. Of course, as I'm sure you're all aware, refractive disorders are probably the most common visual disorders, and these are associated, for example, with the shape of the cornea, but also with the flexibility of the lens. So the classic example of that is that as people get older, the lens becomes less flexible and their near point, i.e. how close an image can be to the eye and still be in focus, gets further away. And so you'll see elderly people who hold a book further and further away from their eye in order to get it in focus. And of course, the response to that is to wear reading glasses, which add an additional level of focusing power. As I said, the image is brought to a focus on the back of the eye, and this is the retina, where we have the sensory receptors. And in the next slide, we can see that image of the eye and two different elements of that. On the bottom left, we can see an image of my own retina, which shows two different areas highlighted: the optic disc, which is the area where the nerves and blood vessels enter and leave the retina, and an area called the macula, which is the area of highest sensitivity, and within that there is a region called the fovea, which contains the highest density of photoreceptors, the rods and cones that respond to light. And it's the fovea that gives us most of our central vision and certainly most of our colour vision. On the right hand side of this image we can see the different cells organised in different layers that form part of the initial processing of the visual image. So a significant part of the processing takes place within the retina itself. This is a complex process, but we're just going to consider the output, which will come from the cells called the ganglion cells. In the first lecture I talked to you about the process of transduction, which is the mechanism by which sensory stimuli are converted into neural signals. And this is summarised in this next slide, where we can see the light stimulus that acts on the photoreceptors, the rods or cones, and within the photoreceptors is a specific pigment, the photopigment, called rhodopsin. And when light is absorbed by rhodopsin, it changes its chemical conformation, and that in turn sets in train a set of enzymic cascades of internal messengers which lead to the closure of sodium channels. Now that's something that might come as a surprise. In the dark, the sodium channels are open and the cells are depolarized. In the light, the sodium channels close and the photoreceptors hyperpolarize. And this is at odds with all the other sensory mechanisms that we talked about, where typically you might expect a sensory stimulus to lead to opening of sodium channels and depolarization. In the eye, we have the reverse. So the cells hyperpolarize and reduce transmitter release. The next slide shows a summary of the process. So the rods and cones hyperpolarize. The signals are processed within the cellular layers of the retina, and there is an output via the cells called the ganglion cells, whose axons make up the optic nerve that then conveys the signals into the brain itself. The ganglion cells have very specific receptive fields, and these are called center-surround receptive fields. Now, what do I mean by that? If you look at the diagram, you can see that there's an inner circle and then an outer ring around that circle. These are the center and the surround of the receptive field. At rest, the ganglion cell fires at a relatively low rate, and that's shown by the train of action potentials right at the bottom of the diagram. If I shine a dot of light on the outer ring of the receptive field, the firing rate decreases. So it's inhibiting the ganglion cell response. If I shine a dot of light on the center of the receptive field, the firing rate increases. So this is the positive response, the center activation. If I shine a broad beam of light over the whole of the receptor, the firing rate increases very slightly by comparison with the resting state, but not by much. So the center-surround system means that the ganglion cell is responding to contrasting light stimuli depending on where they fall within the receptive field. Now, the visual pathway allows us to see where the signal goes within the visual system. The visual pathway shows us the passage of the neural signals from the retina, from the ganglion cells, through to the optic cortex, the visual cortex, where the signals are processed. So from the retina, the axons travel to a region called the optic chiasm, where there is a partial crossing over of the axons from different parts of the eyes, so that comparable regions of the visual fields from the two eyes are brought together. And this is the process that underlies our binocular vision. It's the bringing together in association the two parts of the visual fields from the two eyes so that they are associated with each other. The optic tract then passes to the thalamus. Remember, this is called the gateway of the cortex, to a specific region of the thalamus called the lateral geniculate nucleus. Further processing takes place here before the signal is then passed to the occipital lobe at the back of the brain called the visual cortex. This final slide shows just some simple examples of initial visual cortical processing. Within the different layers of the visual cortex, there is significant and complex processing of the visual signal, as you might expect. The visual cortex is laid out as a map of the retina in the same way as the somatosensory cortex is laid out as a map of the body. What this slide shows is the receptive field of the first stage processing of a cell called a simple cell. This cell responds to a bar of light. And as suggested, what happens is this cell receives input from a group of ganglion cells which are lined up in parallel, so that if we shine a bar of light on the retina, that bar of light crosses the center regions of each of those ganglion cells. So that will be the strongest stimulus. And in the bottom part of the slide, what I've shown are what happens if we orient that bar of light in 4 different directions. So if the bar of light is at right angles to the receptor field, we get no response, the response is inhibited. And then as we gradually rotate it to horizontal, so that the bar of light is crossing more of the central parts of each of the ganglion cells that feed into our simple cell, then the response increases into that third image, which shows the horizontal bar where we get the maximum response. And then as we further rotate the bar of light, the response decreases again. This is the first stage of processing, and then there is a hierarchy of different types of cell that respond to different shapes of stimuli, and indeed also respond to different aspects of the sensory stimulus. So for example, different colours and movement and so on. Within the association of areas of the brain, the visual signal is then linked to memory. So for example, the recognition of shapes, the recognition of faces, and so on.